Hello children, how are you fine? Welcome to my video class. We are going to take up your first chapter in biology of your class 9 NCERT textbook, that is the fundamental unit of life. Now before we start this topic, let me ask you children, if you want to construct a building, what are the basic materials required for constructing the building? You might say, sir, we require brick, we require cement, we require sand. Of course, you are very correct. But what is the basic requirement without which you cannot construct a building? Yes, you are correct. That is brick. So if I make a simple structure combining this brick, adhering few bricks together, with adhering substance, cement. These are one individual brick. In that way you can combine so many bricks to get a building. Am I right? So this individual unit which require for construction of a building, what do you call this? Unit of building. Unit of building. If you consider this building and simultaneously if you consider the body of a living organism, either if it is plant or animals, a human being, or even a very small unicellular organism. So in our body also such types of building block is there which constitute the whole body. That building block, that unit of our body, what do you call it as? Cell. This cell is not only the structural unit of our body, but it is also the functional unit. So, very simple way, how can you define a cell? Cell, it is the structural and functional unit of living organisms. So you got a very clear concept about cell. What is cell? Cell is the structural and the functional unit of any living body, any living organism. So it is the most fundamental unit of our life. Now before we discuss detail about cell, let us have an idea about who first discovered cell and little bit with what type of structure the person who discovered saw under a very simplified microscope during that time when it was discovered. Let me give a clipping. So you see this gentleman, Robert Hooke. Robert Hooke, in the year, as per your NCERT book, it is written here around 1663 in NCERT textbook. It is written very clearly, Robert Hooke discovered the cell with a thin slice of, this is a slice of a cork. Cork is nothing but the bark of plant. So it is a plant tissue. This small compartment, small area you can see, this is thin slice of a cork Robert Hooke taken, and in the year of 1663 with his very simplified microscope. This microscope he has used for observing the thin slice of the cork, and as per your NCERT textbook, again I highlight this point, as per your NCERT textbook, it is 1665 Robert Hooke discovered cell by taking a thin slice of a cork. If I show you a very magnified view of that cork, the cells which is clearly visible, this is a magnified view. Here the cork slice, thin slice, the compartment, that's what you call cell. Here you see the cells, individual structural unit, and this is also the functional unit as discovered by Robert Hooke in the year 1665. Later on, so many modified, improved form of microscope came, and further observation, further the study of different components of cell will be discussed. This scientist Leeuwenhoek, you see in my clipping, he with a little improved microscope, of course a simple microscope, saw for the first time the individual isolated cells from the water body, and also further study of individual cells after the discovery of Leeuwenhoek's individual cell. So let us have, this is the free living cell which was observed by Leeuwenhoek. This is white blood cell, a red blood cell without a nucleus, the bacterial cell, this is the human sperm cell. So this is the individual cell, not as a whole within the tissue, but a separate individual cell identified by Leeuwenhoek. If we progress further, you see this clipping. The scientist Robert Brown, what for he is famous? You might have read in junior classes. Robert Brown for the first time discovered nucleus within the plant cell, this round, pointed, spherical structure present within the plant cell. In the year of 1831, this Robert Brown discovered this structure and he called it as a nucleus. Further we have proceeded, we have got a little more development over the discovery of cell, and later on the cell theory came. Cell theory was given by the two famous scientists, Schleiden and Sewon. Schleiden in the year of 1838 and Sewon in 1839, and jointly their discovery about the cell, that is cell is the basic structural and functional unit, and all living cells originate from pre-existing cells. This was the cell theory given by Schleiden and Sewon in 1838 and 39. Further, the cell theory was more elaborated by other scientists, but basically for the first time, cell as a structural and functional unit, and all the cells originated from pre-existing cells were given by Schleiden and Sewon in the form of their cell theory. Let us now proceed with this cell. If you see the different cells collected from different parts of the human body, this is the muscle cell in the form of a spindle cell structure. This is blood cell, a nervous cell, that is nerve cell. This is bone cell, sperm cell, ovum. This is the fat cells. So these are the different types of cells having different structures within the human body. But if you take a particular tissue, say for example epithelial tissue in animal, say for example epidermal tissue in plant, the tissues are formed of a group of cells having common origin and doing same function. So in a tissue, all the cells are looking exactly alike. Now, let us discuss a little more about the common cells of the human body. Again here, different cells we have shown you, as in previous slide also these cells we have shown. Now, the cells, the organism can be made up of a single cell also, or the organisms, the body of organism can be made up of hundreds and thousands of millions of cells. So, what happens? Where in one hand, certain organisms are made up of only one cell or few cells. On the other hand, certain other organisms are made up of several cells. In the case of those organisms, say for example amoeba, a simplified structure having a nucleus at the center, or say paramecium, having cilia, the macro and micronucleus. These are the two examples of unicellular organism, amoeba and paramecium. This unicellular organism, that means the body itself made up of a single cell, both in case of amoeba, in case of paramecium. And the single cell itself is able to carry out all the functions of their body. On the other hand, if we take the example of we human beings, billions of cells are there in our body. So there is a clear-cut division of labor. Certain cells organized to form one group of tissue. These tissues organized to form organs and organ system. Say for example, if we take an example of human body, so we have epithelial tissue, we have muscular tissue, we have a nerve tissue, that means here this epithelial tissue is made up of so many cells, all are looking alike. Likewise, muscular tissue made up of so many cells, nervous tissue made up of so many cells, that is a neuron. Anyway, when we are talking about cell, in plant and animal, almost the component of cells are same with minor variation. So let us talk about a typical plant cell. Let us take an example of a typical plant cell. I have taken up a figure of plant cell here. You can see, I will draw the same diagram here, a very simplified diagram. It will be helping you how to draw a very simple diagram. In the case of plant cell, you can take a hexagonal or a pentagonal structure. This is the outermost boundary, which is very rigid structure. This outermost boundary, what you call as cell wall. Inside the cell wall, there is a fine, thin covering. You can see with other color of chalk I am drawing this. This is what we call cell membrane. If I take an example of an animal cell, in animal cell there is no such outer rigid boundary, there is no cell wall. What is present here? The outermost boundary is cell membrane. So cell membrane, this structure cell membrane is common for both animal cell and plant cell. This is plant cell, and this is animal cell. At the center of animal cell, or in case of plant, little away from center, there is a spherical structure. This spherical structure, in case of animal cell, almost at the center, there is a spherical structure. This is called the central part or head of any cell. What we call it as nucleus. Very simplified diagram I am drawing for you so that it will give you a very clear understanding. So this is the nucleus of animal cell. This is the nucleus of plant cell. In case of plant cell, there is a specific structure which is present in plant cell only but not in animal cell. That is called plastid. So let me draw a diagram of plastid. It is a double membrane bound structure present only in plant cell. Again I repeat, double membrane bound structure present only in plant cell but not in animal cell. Another double membrane bound structure is there both in plant cell and animal cell, that is mitochondria. What is it called? Mitochondria. Double membrane bound structure. The inner membrane invaginate inside to form a finger-like projections called cristae. So these projections are called cristae. This is the mitochondria. And this is the plastid. Plastid is absent in animal cell, mitochondria in animal cell. Let me draw the mitochondria. Same as plant cell, a double membrane bound structure. Inner membrane, outer membrane is porous. Inner membrane invaginates, draw me like this, invaginates inside to divide to the inner surface into compartments, and this is having finger-like projections. These projections are called cristae. So this is the mitochondria of animal cell. Both in case of plant cell and animal cell, you will find a network of blindfolded tubular interconnected structure. A network of blindfolded tubular interconnected structure. If I draw it like this, which is very close to nucleus. This interconnected tubular blindfolded structures present in both case of plant cell as well as animal cell. Try to draw like this, very simplified form of diagram. You need not be confused. Sometimes book is diagram, so many structures are given. So very simple way you can draw this. This types of blindfolded interconnected structure present in case of both plant cell as well as animal cell. In case of both plant cell as well as animal cell. Only few I am drawing here. These blindfolded integrated structures are called endoplasmic reticulum. What is it called? Endoplasmic reticulum. Let me level it. Endoplasmic reticulum. So this is the endoplasmic reticulum. Endoplasmic reticulum. This present in both case of plant cell and animal cell. In case of both the cells, you can find so many other structures. Very commonly, it's called Golgi body. Golgi body, the name itself, you can remember the most famous scientist Camillo Golgi. He, for the first time, discovered and coined this structure as a separate individual body, what he call as organelle, and in honor of his name, it has given a name Golgi body. Golgi body are again vesicular structures looking like this. Looking like this. Very simplified diagram you can draw in your exam also. This type of diagram if you draw, it will be very easy for you to draw within less span of time. So these are the Golgi body. In plant cell also we can draw the Golgi body here. We can draw the Golgi body. In case of plant cell, a very big structure present almost center or near to the center of the plant cell, what is filled with sap. This structure we call it as vacuole. We call it as vacuole. In case of animal cell also, you can find the same structure vacuole, but comparatively small in size. The basic difference you can see in case of plant cell vacuole and animal cell vacuole. Animal cell, the vacuoles are quite small and few in number. Whereas in case of plant cell, the vacuole is much bigger and generally single in number. Almost in certain plant cell, it is almost occupying the entire area, leaving other component at one side. So within the membrane, that membrane what we call the limiting boundary of animal cell and plant cell, it is having outermost cell wall and inside cell wall. The boundary called cell membrane. In the case of animal cell, the limiting boundary what we call as cell membrane, otherwise called plasma membrane. Now, if we make a comparison of these two cells, you can easily find out the component which is present in plant cell and which is absent in animal cell. We shall come in detail little later. But before that, what I here want to tell you, the limiting boundary covering area within which a central part what is occupied by nucleus, and outside the nucleus within the cell membrane, the area what you call as cytoplasm. So this area covered by a fluid filled cytoplasm. And so here this is the cytoplasm. Within the cytoplasm, so many unified, these are the unified structure. Each are having different shape and different functions. So these unified structures are again structural component of cells. If a cell want to function perfectly, this unified structure must perform their specific function. This unified structure what you call as cell organelle. What is it called? Cell organelle. Cell organelle. So the cell organelles, few cell organelles as I have drawn are mitochondria, Golgi body, plastid in case of plant cell, endoplasmic reticulum. So this, if you see my clipping, here I have shown you the plant cell where the Golgi apparatus, commonly all the structure together is called Golgi apparatus. The individual Golgi bodies or Golgi apparatus. This is the nucleus. Nucleus, I shall show you separately what are the components of nucleus. Then here we have shown the plastid. Then mitochondria, the mitochondria as I have shown here also. Then certain other structures, small structures, peroxisome, etc. These are also present in the cells. So let me show you a clipping of animal cell. This clipping I have taken for animal cell. Here also see the mitochondria, Golgi apparatus, nucleus, endoplasmic reticulum. This is mitochondria. Then maybe some microtubules are there. Centriole, another structure present only in case of animal cell. I will show you this structure, centriole. This centriole, on course of cell division, it divides into and helps the cell to divide. This is a centriole, present only centriole. Centriole. This structure present only in case of animal cell but absent in case of plant cell. Now, if you see the structure of plant cell and animal cell, you can easily compare what are the structure present in plant cell, what are the structure present in animal cell in common, and what are the structures specifically present in plant cell but not in animal cell, or what are the structure present in animal cell but not in plant cell. Just tell me, outermost, what is it? Outermost boundary, rigid structure, that is cell wall. Where it is present? In case of plant cell only. Do you find any cell wall here? No. But what about cell membrane next to cell wall from inside? Inside the cell wall, in case of plant cell membrane. You see the cell membrane here in plant cell also, in animal cell also. Nucleus in plant cell also, in animal cell, plant cell also, in animal cell also. Back wall in plant cell also, in animal cell also. But what is the basic difference? In plant cell, the back wall is bigger and few in number. In animal cell, it is much smaller and more in number. In animal cell, you see the endoplasmic reticulum. Here also endoplasmic reticulum. Plastid is present only in case of plant cell, but not in animal cell. That is why we are not able to prepare our own food, as the plant can prepare during daytime by trapping the sunlight. If the same mechanism had been there in our body, we would have not roam here and there for in search of food and grumble for getting food. Easily we stand outside in sunlight and just would have. But unfortunately, we do not have plastid. This plastid is the site for trapping the sunlight in plant, and that is why they are able to prepare their food. Now, let us discuss little bit detail about one by one. Shall I have it? This part I will have it, and I will take out the individual component of cell. So you see, both in case of plant cell and animal cell, one boundary, that is cell membrane, is common. So how does the cell membrane look like? If you take my clipping here, this is the structure of a cell membrane. This is called a lipid protein bilayer structure. Otherwise, it is also given a very common name in higher classes. We will study in detail about this structure, what you call as fluid mosaic model. Anyway, you need not go in detail about fluid mosaic model in your class 9th standard, but at least you try to understand this outer boundary of animal cell and the same boundary present inside the plant cell, what you call as cell membrane or plasma membrane. It is having two layers. That is why two layers of lipid. It is called as lipid bilayer. If I draw the diagram like this way, this is upper lipid layer having a head and a tail, carboxylic part, tail part, and lower layer. So lipid bilayer having head protruding outward, head part is protruding outward, and the tail part inside. So this is the lipid bilayer, and within this lipid bilayer there presents protein. Certain proteins are present inside, certain proteins are protruding out of the lipid bilayer heads. These are called intrinsic or extrinsic protein. Anyway, you remember simply protein. So certain proteins, I will use some other color chalk. These are the protein. These are protruding outside. Certain proteins are present inside. and it is this protein, some specific protein forms a protein channel which helps for active transport through cell membrane. This is the protein channels which helps for active transport. Anyway, what is the function of this cell membrane? Cell membrane not only acts as a limiting membrane for animal cell, in case of plant cell it present inside the cell wall, but it acts as a selectively permeable membrane. I have used the term selectively permeable membrane. So let me write here, selectively permeable membrane. Why it is called selectively permeable membrane? That means this cell membrane does not allow everything to pass through it from outside to inside or inside to outside. It is very selective in to whom it will allow to pass, to whom it will not. That is why it is called a selectively permeable membrane. Say for example, the various gaseous substances like carbon dioxide, like oxygen from outside can pass through the cell membrane inside or can pass from inside of the cell to outside. Whether it will pass from outside to inside or inside to outside, that depends upon the concentration of the gas present outside or inside. In comparison to outside, if the concentration of that very gas, carbon dioxide or oxygen, present more inside, then from inside it will go to outside. Or vice versa. Same for case of oxygen, same for case of carbon dioxide. So this process of movement of gases through selectively permeable membrane, here we are talking about the biological system. We call it as diffusion. Diffusion. This term diffusion you will use in your chemistry also, chemistry part of your class 9 syllabus also, frequently we will use. Water molecule also enter through this selectively permeable membrane from outside to inside, water molecule, or from inside to outside. Here also this movement of water molecule in both the direction, in which direction it will move, that depends upon the concentration of water molecule. If it is more outside, it will pass through selectively permeable membrane to inside, or more inside the cell, it will pass through the membrane outside. This is also a process of diffusion. But in case of water molecule, we use a terminology called osmosis. Osmosis. So this cell membrane permeates the process of diffusion and osmosis. Now, as far as osmosis is concerned, let I give you a brief idea about there are various activities are there in your NCERT textbook. So let me take up one simple activity. If you take a potato tuber and cut it into sizable pieces, almost rectangular size, this activity I will show you in your activity class in the form of practical. We are going to perform, take it as shown, and slowly from inside, whatever the potato part is there, we shall remove in such a way that this type of a thin shape of potato cup osmometer, potato cup osmometer can be made. Now, if you keep this potato cup osmometer in a container, so let me use some other color. And fill 1/3 of this potato cup osmometer with concentrated sugar solution. What I have taken here? What I have taken here? Concentrated sugar solution. Highly concentrated sugar solution. By dissolving sugar in water, if we prepare a concentrated sugar solution and put it inside this osmometer, level it with the help of alpine. With the help of alpine, level it so that you can easily understand the next day whether there is a rise in the level or not. Whether there is a rise in the level of sugar solution or not. We have leveled in that. Now, the potato osmometer will keep in another container, keeping distilled water. Keeping distilled water. So this is now the potato osmometer. Let me make a shade. This is your potato osmometer. Inside which we have kept the sugar solution. Outside within the container we have kept distilled. What you have kept? Tell me. Distilled water. Distilled water. Distilled water. You keep it for few hours or say overnight. Next very day when you are coming to your laboratory, biology lab, inspect whether any change of the level of sugar solution within the potato osmometer has taken place or not. For your surprise, you will see the water level or the sugar solution level inside the potato osmometer has increased. What will be the next level now when you are observing after overnight? Say the level might be this or so on. Now question comes, how and why the level of sugar solution increased? It is because the concentration of water inside the osmometer was less. So water concentration less. Less water concentration. In comparison to outside, here the water concentration is more water concentration. Here we have added sugar in water, thereby making this sugar solution. So here the concentration of, and here we have taken distilled water. So the concentration of sugar, concentration of water, as because there is no sugar or no electrolyte or no other substance here, distilled water we have taken. So concentration of water outside the potential osmometer is more, inside less. So what happened? There is a flow of water from outside through the semi-permeable. That's what we have prepared the osmometer by taking as much as removing from inside, making a capsule structure, only a thin membrane left. That is acting as a semi-permeable membrane. Through the semi-permeable membrane of potential osmometer, the water molecule H2O started moving inside the osmometer. Is that clear to you? Why? Because concentration of water is more here, concentration of water is less here. If we use some other terminology, that means outside it is hypotonic. Inside it is hypertonic. So, through the semi-permeable membrane, water molecule started moving from the hypotonic to the hypertonic solution. Is that clear to you? If we take a reverse process of same osmosis, reverse process. If you take a rheolite and take a thin slice, keep the slice over test tube with a drop of water, covering with cover slip, and observe it under a microscope. The structure of cell, suppose you observe like this in an elaborated form. I am drawing here. Suppose a round shape. Now, if you add drop by drop concentrated sodium chloride solution, NaCl solution, sodium chloride solution, drop by drop from outside, and after some time, if you observe this lead pill, what you will observe is there is a shrinkage. Let me use some other color. There is a shrinkage of this plasma membrane along with its content. This is the cell wall. And the structure will look like this. Why it happens so? Because outside we have given drop by drop concentrated high sodium chloride salt solution. So inside, within the cell, rheolite cell, the water was earlier more. Outside here, the sodium chloride solution, salt solution having less concentration of water. So from inside the cell, the water molecule will start coming out to the medium. As a result, this cell will now shrinkage of cell. This is because of water molecule coming out. What is this? Outside, this is the cell wall. Cell wall is a rigid boundary That is why there is no overall change of the cell size, but the cell membrane along with its inner content there is a shrinkage because of through this cell membrane the water came out. This is called exosmosis, means osmosis from inside to outside. Earlier we have given the example, here there also the water molecule, but from outside medium inside the cell. Another example, very exciting example in your day-to-day life. If you take a glass tumbler, today you do this activity, very simple. You take glass tumbler, half filled with water. All of you today itself you try to do it and put few grains of resin, dry resin, which is almost like the structure dry resin. Overnight you allow it to keep it as such so that the endosmosis can take place. Next very day, if you observe, what you see here, the resins become fully swallowed. The resins, now this is the structure of resins. Why it is swollen? It is because water molecule inside the resin, earlier there is highly concentrated form of sugar, almost negligible amount of water. But outside there is full of water. So from outside the water molecule will start entering through cell wall pores via cell membrane. Acts as a semi-permeable membrane. Through the cell membrane, it will enter to the cell protoplasm, and gradually due to entry of water, there will be a pressure built inside the cell membrane, and cell membrane will go on increasing. But of course it will not burst because of there is a outer limiting membrane called cell wall. So this resin now swallowed. It is because of the entry of water through endosmosis. What we call it as endosmosis. And due to entry of water, there is increase in turgor, what we call as turgor pressure. That is why the dry resin now fully becomes swallowed. So this is how we have tried to give a concept of what is the function of cell membrane. It is not only acting as a limiting boundary, but it is very important because it is through this membrane certain substances can enter inside the cell or go out of the cell. That is why it is acting as a semi-permeable membrane made up of lipid bilayer along with protein, intrinsic protein, and along with certain carbohydrates. These proteins are also making protein channels, and these protein channels are very important for active transport. Of course, there is no scope of discussing what is active transport here and how it is taking place. We shall discuss in some higher classes. But for the time being, you try to understand these protein channels are very important through which the active transport by using energy are taking place. So I hope whatever I have discussed today, a brief I have given about cell, the cellular component. Later on in my next class, I will discuss in detail. I will take up one organelle, their detailed structure and functions. So whatever I have discussed today, I believe that you got a very clear concept. Till then, bye.